All right, you guys, welcome to this week's episode. This little quick video on Meselson Stahl experiment. So here you are. You should be in DNA replication and Meselson Stahl experiment right here. So you're almost done with DNA replication. I don't have a quiz or a video or a test or anything on this. I just want you to end off with this little assignment, and that's how I will end it in my class. So here we go. So with this video, you guys, basically what we're going to be doing is looking at this driving question. Which model correctly shows how DNA replication occurs? And so when we're looking at this, you guys, at this time, just to kind of give you guys a little refresher, remember Watson and Crick have now discovered the structure of DNA. And with the structure of DNA, they had assumed that the type of the way that DNA replicated was through semi-conservative model. Okay, and we're going to look at that a little bit. But I really want to look at the experiment that was carried out to really support and show that, yes, it is the semi-conservative model. Okay, and who did this was a little bit after they discovered the structure of DNA. There was a couple scientists, Matthew Meselson and Franklin Stahl, that conducted this experiment. Okay, now this experiment, it's not too hard to understand what they did after, and so I'll kind of explain that. But what I want you to do first of all is to pause the video, and I want you to read the introduction really quickly. Okay, I also want, and so stop before testing hypothesis. So I want you to read this part up here. And so pause the video. I'm going to wait for a second as you pause, and then I'll start going again. Alright, just took a little drink of my Mountain Dew here to keep me going during this coronavirus situation. Okay, so here we go. So if you guys read the little this introduction, I'll kind of explain this experiment. So with this experiment, again, at first they thought there might be three different possibilities of how DNA gets replicated. So the first one was the idea that this semi-conservative type of model. The idea with the semi-conservative is that here, let's say we have one strand of DNA. We know that every time this cell has to replicate and duplicate itself, it needs to make duplicates of the DNA as well. And so we can see with the semi-conservative that each strand forms as a template to make a new strand, which is shown in blue. So purple is the old strand, blue is the new strand. Right here we can see the exact same thing happening, where one strand becomes a template to make the new strand, and they stay together for the second or for the first generation. So here's where we start. After one round of replication, we see this. And then if we were to model this again to go to the second round of replication, we see that again the same thing occurs. The purple will become a template for this one, and then the blue strand that was new for this round will be the template for this one to make another new strand. Okay, and we can see that happen, and we could model this forever and ever, right? With the conservative model, it was kind of similar, but a little different in a way. And this is what I want you guys to understand, the differences between these three. So with the conservative model, each strand forms or acts as a template. However, with the conservative model is that both of these old original strands will always come back together, and the new strands form together. Okay? With the dispersive model, this one's kind of strange, but it's the idea that it's chunks. So you can see the bits and chunks are embedded throughout in the molecule. Okay? So with these three different models, they were like, okay, well, let's set up an experiment so we can truly see which one of these is correct. And so what Meselson, Stahl, and Matthew Meselson and Franklin Stahl did is they took bacteria and they fed bacteria. They knew that on our bacteria are nitrogen, so we can change the weight of our DNA molecularly by changing the type of nitrogen that we place on that DNA. And so you can see that he had two types of nitrogen. So these are two different isotopes of nitrogen. We had nitrogen 14 and nitrogen 15. And he knew that nitrogen 15, since it's an isotope, it's got an extra neutron in the nucleus, and so it's going to be slightly heavier than the nitrogen 14. And he was like, okay, well, by doing this, I could put DNA on a test tube that's filled with what we call cesium chloride. Now, the cesium chloride will act as a gel. And what will happen is when they place the DNA on top of this test tube and they put it in it's called a centrifuge to spin it, and it'll spin it very, very quickly. And as it spins it, it will gravitational pull will pull the DNA through the cesium chloride. The heavier the DNA, the further down it goes. Okay. So he was like, okay, well, let's use this type of technique to test which one of these is correct here. Sorry for the sound. Anyway, so here we go. So what he did is he took some E. coli, some bacteria, and he fed the bacteria a rich diet of nitrogen 15. Okay, and he let it grow for several generations to ultimately lead to that all the DNA inside of that bacteria was all nitrogen 15. To test this, he then would place the DNA from the bacteria in the cesium chloride, and what he was able to find, and what he did is, let me model this for you. He found that there was one band of DNA, okay, and it was down here at the bottom. So showing that there is a heavy band of DNA found there at the bottom there, okay. So then he was like, okay, so since I have it all nitrogen 15, it's all very heavy, let's test and see, let's change the type of food source, and let's change it to a type of 14, nitrogen 14 in the culture, in that E. coli. And let's test and see how the DNA changes every time it replicates. So kind of an interesting procedure there, a brilliant procedure. And so with that, we can then really see, well, how is it changing? Okay. Do we always see one strong, heavy band and some light bands? Do we see a heavy band, then a medium-sized band, and then medium and light? Okay, and so we'll model this for us here. Okay, so here's the first part. So the first part I kind of just gave you the answer for that. It just wants you to get a colored pencil. Now, you guys, you can do this all online. So you can like get this little, I'm actually interested to see if this editor, nah, that's not it. Okay, so you guys, a couple things that you can do here. You can't really highlight text on this, but if you wanted to, you could copy these pictures, okay, and then go to Paint, and using Paint, you can draw and do all the things you need to, and then just re-upload it into this where the picture should be, okay? So you guys, you can easily put these things in there and alter it on the computer. Now, let's say that you're doing this on like an iPad or things like that. The awesome thing is that the Word version of that's found on your iPad or your phone, if it's an Apple product, is up here in the center. There will actually be little pens, and you can actually draw right on that. Okay, so manipulate this the best, however you need to show me the data and get the full points on this. Okay. All right, so here we go. So let's do this first one together here. So it says, if DNA replication is semi-conservative, boom, whoa, is semi-conservative, illustrate the arrangement of light and heavy isotopes of nitrogen on the DNA molecules formed in generation 1 and 2. Assume that each bacterium divided exactly once per generation. Okay, so we're going to sketch that here first, and then I will then we're going to see what it would look like on cesium chloride. So what I'm going to do really quickly is in red, my red will be my nitrogen 15. Okay, and then in blue will be my nitrogen 14, so my new bands. All right, so let's do this first. So we know we started out with red DNA, and that's for every single one of these that you guys will do. Okay, and we know that for one generation, what would then happen? So here we are, that it's all nitrogen 15, but now they have a nitrogen 14 source with the semi-conservative idea. And if you need to, refresh and look back at that top picture. But we know that each of these will form a template to make the new band of DNA. Okay. So here we have our DNA, and I could, If I wanted to get intense there, I could do it like that. Okay, so anyways, I've got my DNA, and you can see that each one formed a template, and then to make that new band of DNA. Well, what's going to happen for the second generation? Well, we see each one of these would then form a template. I'm just going to draw those like that. And then my blue will be formed right there. I also have this blue one that's going in there to make a new blue, and like so. Okay, and right here. Okay, so now I've got my DNA form. Now the idea is, well, how could I then place that on my cesium chloride test tubes? And so looking at generation 0, what color of band do we have? We have 2 heavy bands. And so where would that be found on the centrifuge? Well, it's going to be a heavy band. It'll be found there at the very bottom. What about generation 1? I want you guys to pause and put it where you think it's going to be. Be ready to erase maybe if you don't get it right. Okay, so maybe you've attempted and you've tried that. So when we look at this, you guys, the key thing, and this is what I've seen a lot of students do, is they want to separate this out. And they'll make, well, we have a heavy band, and then we have a light band, because we have 2 lights there. No, that is not correct. Erase that. Okay? This DNA cannot separate. Okay? It cannot separate. It's kind of half and half. So what we actually see is a band right here in the middle. I'm going to do that in purple since it's a mixture between blue and red. Yeah, I can do colors. I'm an artist too. So anyways, we have this medium band there in the middle because both of them are kind of medium. Okay, well, what do we see right here for generation 2? Well, I see this band is this medium purple color. So is this one. Then we have 2 light. And so I'm going to have the same thing going on right there. But am I done? No, I'm not done. Because I actually see a light band up here at top from these 2. And then this one forms that, and this one forms that too. Okay? So this is kind of the pattern that you would suppose to see from semi-conservative. Now, you guys, I want you to do the other ones by yourself. Okay? And I'm not going to help you. But using this same idea that I just did, you guys can do it. Okay? And so looking up, missed some parts there. There we go. Okay, so the next one, you'll look at it, what if it's conservative? And then the last one, what if it's dispersive? And then what you'll do down here is you'll actually analyze this. This is actual footage data. This is actually the picture data that they got in their different test tubes. Right here is showing, I would specifically look, you guys, at this one right here for generation 1. Then this one right here for generation 2. And find the one that matches this. Okay? Now generation 0, if we had it right here, it would be there would be a band like right in here. Okay? This is showing heavy. And this right here is showing light DNA. Okay? So use this to help you answer that question. And so what you guys will do to turn this thing in after you've filled all that out, down here you'll do my famous CER. Okay, so find a claim. Which type of model is accurate? What data and evidence do we have to support that? And then reason this out. Also, you guys make sure when you reason this out, you pick a cross-cutting concept, as we've discussed. So, what cross-cutting concept might help you to support your claim? Is it patterns? Is it cause and effect? Is it structure and function? Pick one and then explain that, and you can just type that right in here. Okay, and then kind of conclude, pull this all together into a final summary for you there, and turn this puppy in. If you guys have any questions, you guys, please don't hesitate to let me know, okay? I know this is a challenge. It's a challenge for me. I miss you guys so much. I wish I could just be there in front of you guys, bugging you and helping you along this step of the way. It's been a real challenge for me as well to have to teach online full time. Okay, but no, I'm here for you. I'm just a phone call away if you need to. email me, whatever is best, and I will be more than welcome to help you through any of this stuff.